Friends, today we see the control of nitrosamine impurities in human drugs. Guidance for industries. It is published by the Department of Health and Human Services, Food and Drug Administration, Center for Drug Evaluation and Research (CDER), September 2024. There is a pharmaceutical quality and manufacturing standards. These are a table of contents. Then control of nitrosamine impurities in human drug guidance for industry. Then introduction. The guidance recommend the states manufacturing applicants for active pharmaceutical and APIs. Drugs products should detect, prevent, and acceptable levels of nitrosamine impurities in drug products. This guidance recommended for the states manufacturing applicant of powerful pharmaceutical ingredients and drugs products should take into detect and acceptable levels of nitrosamine impurities in drugs products. The guidance also describes the conditions that may introduce nitrosamine impurities. The unacceptable handling of nitrosamine impurities, which are probable or possible human carcinogens, in certain drug products, as may clear the need for a risk assessment or strategy for the potential presence of nitrosamine in any drug product. The discovery of nitrosamines in some drug products led FDA and other international regulators to conduct a detailed analysis of these impurities in affected APIs drugs products based on the agency's current understanding. The guidance discussed the potential root causes of nitrosamine formation, advises API and drug product manufacturer applicants that they should use the three-step mitigation strategy described in the guidance. One is the conductor's assessment for approved and marketed drug products and with pertaining applications to a perform. First one is the conduction of assessment of the approved and products and pending with application for approval. Then second is the confirmed testing of risk is identified. Third one is report changes to prevent reduce nitrosamine impurities to FDA in accordance with the applicable requirements. Most importantly, API and drug product should take appropriate action to reduce the prevent the presence of nitrosamine impurities in API and products. Although nitrosamine impurities have been found in some drug products, these have been some recalls to batches that these products have been where unacceptable levels of impurities. Uh, Nitrosamine impurities might exist in other APIs and drug products due to use of vulnerable processes materials in various nitrosamine impurities. Therefore, the recommendation of main guidance applies to all chemical synthesized APIs. Drugs products containing chemical synthesized API and fragments. Drugs products risk due to other factors described in these case guidelines. Semi-synthetic and fermentation products that are risk due to their structure similar to chemical synthesized API. The guidelines applicable for the chemical synthesized API drugs products containing chemically and synthesized APIs. Drugs products at risk due to also this guidance. Semi-synthetic and fermentation products at risk due to their structures and similar to chemical synthesized API. These guidelines this guidance for the same and issued by these are revision of a guideline issued in the February 2020. Among other changes, this revision introduced new section of describe the nitrosamine, dust safety and related impurities, NDSRS, potential root causes of NDSRS, migration strategies or prevent or reduction of presence of NDSRS. With this revision, this guidance describes two general structures class of nitrosamine bodies, small molecule nitrosamine impurities that do not share structure similar to the API and found in many drug products and NDSRS are similar to the API as generally unique to each API. This guidance describes the two categories of nitrosamine impurity. First one is small molecule nitrosamine impurity. That's other in the NDSRS, which are similar to with the API. In the guidance, FDA explains the purely and highly technical nature of the relevant information. FDA intend to provide certain updated information in connection with the updated guidelines of the FDA page. These include the updated information about the recommended acceptable intent for the nitrosamine impurity, recommended safety testing method of nitrosamine impurities and recommended analytical evaluation method for nitrosamine impurities are described in more detail below. FDA also intend to provide updated information in connection with these guidelines of the same FDA press, including the recommendation and the other emerging scientific and technical issues and recommended timelines. This associated information will be updated periodically. In general, FDA's guidance documents do not establish legally enforceable responsibilities. Instead, guidance describes the agency's current thinking on the topic should be in the recommendation. And background is FDA has been investigating the presence of nitrosamine in certain drugs products since 2018. FDA has investigation for the nitrosamine since 2018. And nitrosamine have been found in the search as a angiotensin 2 receptor blockers, histamine 2 blockers, anti-diabetic medication, then example antibiotic rifampin and rifampicin, smoking cessation medications such as varnicellin. FDA continues to learn a system of nitrosamine impurities drug for various drug classes. Is working with manufacturers and applicants to assess the products, their products and determine the appropriate actions. Based on the nitrosamine impurity issues to beyond their just supply. FDA and regulatory authorities have threatened with certain information coordinators, inspection efforts, coordinated efforts, and analytical methods to detect, identify whether nitrosamine impurity develop in a rapid solution, ensure the safety and quality of drugs products. Nitrosamine impurity is cause of impurity. Nitrosamine impurity. The term nitrosamine describes the class of compounds having chemical structure of nitrous group bonded to the amine. This other class of structure of nitrous group amine has been bonded to the amine. The compounds can form by a nitrocycling reaction between amines, that is secondary tertiary quaternary amine nitrous acid. It is nitric acid under acidic condition. A different precursor of 1,1-disulfide hydrogen can be oxidized to form nitrosamine. The compounds once actively bonded on nitropropylen and methyl nitropropylen has been formed via hydrogen oxidation process. Nitrosamine compounds are potent generators agents in several animal species. Some are classified as probable human carcinogens by the International Agency of Research on Cancer. They are reported as compounds in the International Council of Organization of Technical Requirements for the as per ICMC, nitrosamine cause reported as a chart of concern. Compounds in the International Council of Formulation of Technical Requirements for the Pharmaceutical Use. Guidance for the industry. MC1R2 assessment and control of DNA determinant including pharmaceutical to limit potential carcinogen risk. Recommend the control of any non-mutagenic carcinogen and nitroso compound below the level that will be eligible human cancer associated with the exposure to compound. That's small molecule number. Several small molecule nitrosamine impurities could be present in API. Drugs products including the nitroso, dimethylamine, NDMA, NDEA, then NMPA, NIEPA, NDBA, then NMBA, then NDSRH. An NDS is a class of nitrosamine sharing chemical structure with the API or API fragment or the chemical structure are generally unique to each API. NDS is formed to the nitrogenation of API that is such as a nitrate impurities in the excipients. Then a representative reaction API containing secondary amine functional group is structure or acidic condition. Root causes of presence of small molecule nitrosamine in the API. Information gathered by FG suggests that several general root causes of the presence of nitrosamine in impurity. General conditions that lead to nitrosamine impurity. General conditions that lead to nitrosamine impurity formation in the API is the formation of nitrosamine is possible in the presence of secondary, tertiary, quaternary amines and nitrite salts under acidic reaction conditions. Under this condition, nitrite salts may form the nitrous acid, which can react with the amine to form nitrosamine. These are the greater risk of nitrosamine formation if nitrous acid used in the quench residual acid. Use of tetrazole ring formation, introduction by the functional group into molecule in the presence of precursor amines. Nitrates and all as used as reagents in the one step can carry over the another substrate despite purification operation react with the amine to generate nitrosamine impurities. Therefore, whenever nitrate salts are present, carry over into subsequent site can be pulled out in general process that use Actors in the presence of secondary tertiary quaternary amines are a trace of generating nitrosamine impurities. Sources of secondary tertiary quaternary amines that come from nitrosamine amines may be present in the manufacturing process at a variety of reasons. The API intermediates and API raw materials may contain secondary or tertiary amine functional groups. Tertiary or quaternary amines may also be added intentionally as reagents or catalysts. All these types of amines can react with the nitrous acid, other nitrosating agents to form nitrosamine. Amide solvents, such which are susceptible to degradation under certain reaction conditions, are another source of secondary amines. Amide solvents, which are susceptible to the degradation under certain reaction conditions, another source of secondary amines. For example, then formation of ND may form ND. The example is yarn. Then a vendor source raw material containing nitrosamine impurities. Nitrosamine impurities can be introduced in the vendor source raw material contain nitrosamines or precursors. The agency has observed that the following root causes of nitrosamine impurities in vendor source material. This is a nitrosamine amine found in a from cell solvent, orthoxylene, toluene, methylene chloride, which impurities were carried over the drug transfer between storage vessels using a shipment of solvent. Sodium nitrate is known as a non-impurity in some starting materials and may be present will react with the amines under acidic conditions. There is nitrous contain raw materials such as potassium nitrate, then contain nitrate impurities. The amount of nitrate impurity can be tolerated. Process dependent and should be determined by API manufacturer. Then second or tertiary amines have been reported as impurities in some raw materials, as in freshwater as I told you. Then API starting materials, API intermediates may be reached through cross contamination at the manufacturing site. If these materials and manufactured nitrosamine impurities can carry over from the other process. Awareness of the supply chain of the API raw materials in the important factor is preventing in the nitrosamine impurities cross contamination. Awareness in the supply chain is important for the prevention of formation of the nitrosamine in the air. And then third one is the record solvent reagent catalyst as a source of nitrosamine impurities. Records materials such as solvent reagent catalyst may cause risk of nitrosamine impurity due to the presence of residual amines. If recovery processing was the functioning step in nitrous acid using the decomposition of the residual acid, nitrosamine could form during the solvent recovery. Thus, nitrosamine may be entered if they have boiling point or solubility properties similar to the before metals. Depending on how recovery and subsequent prevention take place, this further increases the risk of nitrosamine in the material recovery for these reasons. Some drug products using the API manufacturing by certain low-risk processes were found contain nitrosamine impurities. The agency has observed the following due to root causes. The manufacturing site may produce the same API more than one synthetic process that uses common solvents. If any synthetic process produces nitrosamine or condense in precursor amines, the solvent sent for recovery at risk. There is a record solvents that are contaminated from different processes across the manufacturing lines without control manufacturing can introduce the nitrosamine impurities. If a record solvent with nitrosamine impurities are used in API, the API will contain the impurities even the synthetic is not mandatory. Now we set it about nitrosamine formation. Then recovery of the raw materials. That is a solvent, reagents, catalyst are open or sourced to the third party contractors. Process outsourcing can pose a risk of third party recovery. Facility does not receive enough specific information on the contents of the material it is processing by responding on the routine recovery process. Raw materials can contain nitrosamine impurities. Adequate cleaning between the customers between the different materials is not carried out, is not validated as capable of removing the each impurity of concern. If cleaning method is not adequate, then may contain carry over the nitrosamine impurities. It was reported that nitrosamine would have into the recycle also xylene, then toluene to the inadequate Then use of the shared storage delivering customer identity with another recipe procedure can lead to cross contamination if precautions to our nitrosamine is not placed before the materials from different customers are combined for the recovery. The nitrosamine is included in lot sort of categories and butyrene chloride third-party control possible to the combine catalyst lots from the multiple customers. Pinching procedure for the nitrosamine impurities. The risk of nitrosamine formation when the pinching stage is conducted directly in the main reaction mixture. When nitrous is added to the reaction mixture, become the residual acid, they are able to nitrous acid to come into direct contact with the residual amongst the raw materials used in the manufacturing process. The nitrosamine impurities could be carried to the subsequent adequate removal of the purification operation not in place if the enter the downstream process once they are introduced. Even the pinching procedure conducted outside the main reactor. These are risk of recovered materials containing nitrosamine are introduced in the main process. Lack of process optimization and control. Another potential source of formation nitrosamine is the lack of optimization of manufacturing process or reaction conditions such as temperature, pH, and sequence of additive reagent. Intermediate salons are inadequate brewery control. Ability has been instructed this reaction condition between the batches and even between the different processing companies and facility of API. Then nitrosamine included in the drug product from the sources other than API. The nitrosamine containing the API other than the API sources for the nitrosamine impurity formation. Nitrates are common nitrosamine agent that have been reported in many experiments in parts per million levels. Nitrate are found in a range of common use experiments which may lead to nitrosamine impurities forming in drugs product containing the drug product manufacturing process saved like storage period. The manufacturer or supplying qualification of program should take into account of nitrate involvement vary across excipient lots may vary by supplier, drug product manufacturer, and applicants should also be aware that the nitrate and nitrosamine impurities may be present in potable odor. Additionally, when nitrosamine precursors such as secondary dioxide quaternions using API may exert impurities in the drug substance. These precursors can react with nitrosamine or exponentially nitrous from another source of aid in the manufacturing process are still molecular nitrosamine NDS are in the products. Some contain a process history, including secondary packaging company manufacturing equipment could be source of nitrate and nitrosamine impurities. These impurities may leak into the drug product containing manufacturing conditions molecular nitrosamine nitrosamine. The risk of such impurities should be assessed during external and reachable studies. Root causes of breakage of NDS in the drug product. NDS can be generated during the manufacturing process, storage of finished product, or is silk type. The non-root causes of the predictive NDS in the drug product are nitrosating impurities such as residual nitrate in excipient. Other sources of nitrate impurities present in the drug products with nitration and active ingredient to the yield NDS in the certain reaction condition. NDS are carried from the APS. Generally, the presence of high levels of NDS can be associated with the drug product rather than API because NDS formation most often results from the reaction between the API and API present in the drug product. Subsequently, NDS levels may increase the proof of storage drug product containing API to secondary quaternary amines. Then recommendation for control of mitigation of nitrosamine impurities in drug product. Yes, because nitrosamine is probable immune causing everything that the personalized acceptable intake limit. A limit depends on the ICGM, so the level of acceptable increased cancer rate. One of the additional page on the subject based on the conservative assumption in the daily exposure of vitagenetic immunity in drug substance drug product over the lifetime exposure that is 70 years. What is the a limit? Is a limit based on the exposure ICGM. So level of exposure increase the cancer rate one in a additional kg in a 1 lakh patient over the exposure of 27 years. Then FDA recommends the limited time using the predicted carcinogen potential as an output with the robust carcinogenicity data. Additional safety information, including bacterial mutagens, the specific nitrosamine compounds are not available. Other approaches are determining element of specific nitrogen database and literature such as available carcinogenic and bacterial we know testing of specific compound. FDA published recommended AI limits for certain NDSRS on the nitrosamine guidance web page. However, unlike small molecule nitrosamine proteins, most NDSRS like history and carcinogenic data, mitigating risk of assessment of the challenging because lack of data nitrosamine guidance web page. Then unrecommended tested medication strategy. API and drug product manufacturers applicants should take following tested nitrosamine included in the drug products. Then first step is assess the risk of nitrosamine impurities on API market products, drug product under the approved or pending application. Then risk assessment should be first one is the conduction of risk assessment. Risk assessment should be conducted timely manner based on the practitioner of the manufacturers. Applicant do not need submit specimen during the agency should be written at the computer required. The second one is the perform confirmatory testing when there is a risk of presence of nitrosamine impurities. Due to nitrosamine physical properties, so much volatility has in analytical test method for the nitrosamine impurities to have specificity, excellent chromatographic separation, highly sensitive detection capability. Report suggests data implemented to prevent the reduce nitrosamine impurities in API and drug products to FDA. This includes submission of drug products, drug master plan in accordance with the 20 CPR. Then recommendation of API manufacturer. First one, mitigation of the presence of nitrosamine impurities in API. FDA the API manufacturers take different stage for the recommended mitigation of API and the API. Then API manufacturing should optimize the design of manufacturing process of API. During route of synthesis, development of many and prevent the formation of nitrosamine impurities. API manufacturing should process contribution ICS M7 for the minimization of nitrogen impurity is possible and avoiding the use of nitrous amide solvent that amide solvent should be avoided where there is unavoidable excesses and cost including the assessing whether nitrosamine can form replacing the nitrous with the other quenching agents. Nitrous should be replaced with the other quenching agents. Optimization and consistently controlling the sequence of reactions process reaction conditions such as pH, temperature and reaction time designing the manufacturing process that facilitates the purge of nitrosamine to substance manufacturing stage. API manufacturing should remove the quenching steps. Then API manufacturing should audit their supply chains. Audit supply chains should be audited monitor them then to any risk repeat of material and intermediates. To avoid cross contamination, we record materials such as solvent and reagents, catalysts should be on and cross contamination should be avoided. Then API manufacturer should avoid a portable water using API manufacturer should contain low levels of nitrates and even the nitrosamine from the environmental sources. Then API manufacturer rework the API batches. The quality students also then repossess was by repossessing revocation should be recommended as per SCS 7. Control of nitrosamine impurities in API. Nitrosamine impurity in the API above the LF. Then API manufacturers should develop a strategy to ensure nitrosamine levels remain below recommended ALM. Manufacturers should develop appropriate control strategy to ensure the nitrosamine levels remains below the recommended AI limit in the API. Taking into consideration best batch variation, even existing uncertainties regarding the nitrosamine impurity their presence in the for at-risk APIs with impurity detected above the 10% of recommended API. Manufacturer should test each batch for the before the release and should conduct the stability studies. Recommendation for product manufacturing applicants, risk assessment and confirmatory testing. Then the code of manufacturing applicant should conduct a risk assessment to determine the potential small molecule nitrosamine input in interaction in the product or risk of assessment of the collaboration of the API manufacturer to aid the identification of APRs. Then processing conditions of API manufacturing should orient the product risk of nitrosamine input. Control of nitrosamine impurities in drug product establishing reliability of API suppliers. Drug product manufacturers and applicants must representative sample of all incoming compounds containing the lots of risk API. Then require the supplementary safer as well as guideline 210. Then establishing a specification of the drug product. If nitrosamine is detected from the about the liquid, the manufacturing and applicant should develop a strategy to ensure the if above the 10% found the nitrosamine impurity, the manufacturer should develop a strategy to include specification limit to the nitrosamine impurity in the drug product. Then complementary testing find levels of nitrosamine impurity we found the about 10% of the including stability studies that expiry. And then control strategy should be established. The recommendation of drug product manufacturing application the reduction and prevention of NDSR formation of drug product. Manufacturing applicant should consider following mitigation strategy when NDSR are detected. The screen excipient the supply collaboration program the occurrence of the potential nitrogen produced across the supplier excipient should reduce the risk of the nitrogen formation is explained. Then design drug products incorporating the antioxidants such as ascorbic acid, vitamin C, ascorbate, salt, alpha tocopherol, propylene glycol. Then modify the microenvironment to natural basic pH during the formation. The formation interaction typically occurs in the acidic condition in the neutral basic environment. The kinetics of the reaction should be considered. Then implementation of recommendation of the assessment of test result. Manufacturer should applicants should be treated mitigation described in the section for the typically comparable is on the sampling of product for specific manufacturer. This other compound presence of impurity shows that it is not present in the alternately. Step one is a if confirmatory testing indicate the nitrosamine levels are more than 10%, recommended AI limit, then specification method analysis test is not needed to project cause of will understood. Then manufacturing process control step after validated. Nitrosamine levels then only the confirmatory testing exceed the 10% of the recommended diameter and within the recommended diameter the control of nitrosamine it should be established to release the stability. Basically it should be tested before the release and tested as per the study patient. If all the upload the product matrix should be improvement implement change being repeated by 30 days. If comparable test indicate nitrosamine recommended element, the formation of manufacturing, the packaging are warranted manufacturer should implement the changes to demonstrate the waste nitrosamine levels the recommended element for product. These changes should be determined as per aesthetic criteria. Then a recommended timeline for implementation discusses confirmatory test is required for the one for the design and drug product development drug treatment. Then pre-submission stage priority prospective applicant should ascertain the risk of nitrosamine impurity as early as possible for development as possible as feasible before performing the even estimate PhDs. The nitrosamine is evaluation should be done in the before the in the development stage. Then after submission the nitrosamine impurity should be uh, tested as per the test decision. Mitigation strategies such as identification of risk and conduction of risks, identified conduction of risk, uh, confirmatory testing, and then after confirmatory testing, third step should be evaluated after submission. After completion, the practical application should conduct the assessment expeditiously and form FDA's confirmatory testing is within a limit. Is approved for the approved product and reporting changes to mitigate that is reporting change by API manufacturing, reporting changes for drug products properly mitigated of nitrosamine impurities, then stable data to support the given guidance with the information. Biostudy evaluation report should be given as per the guideline. Then example given for the requirement of biostudy during the nitrosamine. Then maintaining the drug substance if they can work with the manufacturer applying with the maintaining drug substance. If we nitrosamine impurities identifying the drug products, avoid impurities in the drug supply into the market. If drug product batches are distributed in the certain levels of nitrosamine Only FDA recommended element the API matching also red distribution of drugs of the manufacturing should intimately contact with the drug evaluation research for the API requirement. This right kind software. Thank you.